Hello students, I hope you all are doing good. I Raminder Kaur, your biology teacher. So in this video lecture, I will start with chapter number 10, Microbes in Human Welfare. Microbes, as we all know, these are microorganisms which cannot be seen with naked eye but can only be observed under compound or electron microscopes. Here we will not talk about the diseases caused by them, but we will discuss the useful components of these microorganisms for humans. So before starting this chapter, let's discuss the main objectives behind studying this chapter. So it involves those microbes which are helpful, number one, in our household products. It can be curd, it can be cheese, bread, or many other household dairy products and others. Number second, industrial products. When we talk about industrial products, it means at commercialized level at a large scale. So when industries are manufacturing enzymes, organic acids, wine, beer, whiskey, means different kind of alcohols, that includes industrial products. Number third is sewage treatment. We all know that there are several action plans by which water is being treated with the help of microorganisms. Number fourth is production of biogas. So methane gas is produced in biogas plants. So that is with the help of methanogens. Number five, biocontrol agents. Those microorganisms which can reduce the infection of pest and insect in plants, that is in biocontrol agents. And last is biofertilizers. We all have heard of organic farming. So how can microorganism play a significant role in increasing both quality and quantity of crops? So this we will study in this chapter number 10. So students, let's start. Number first, they have told us that beside the macroscopic, you can write down magnificent. Those who are big, which are easily visible, that is macroscopic. Microbes are the major components of biological systems on this earth. So we have studied in class 11 as our first unit was talking about all the kingdoms. You all remember that first of all, there were only two kingdoms. One, plant, and second, animals. Then with the help of R.H. Whitaker, 5 kingdom classification came. So number first, kingdom Monera, then Protista, then fungi, plantae, and animalia. So students, can you give me this answer? Do you remember which kingdoms among the living organisms contain microorganisms? So your answer is kingdom Monera, Protista, and fungi contain microscopic organisms. Now they have asked one more question. Which are the ones that are only microscopic? So if we talk kingdom Monera, all the different kind of bacteria, so answer is Monera is the only one that has completely microscopic organisms, because there are several protozoans which can be seen with the naked eye also. So they are present everywhere. They are present in our hands, food, water, inside our body, in the sky, soil, in oceans. We can say they are found everywhere. They are omnipresent, right? So not only on these simple surfaces, but even they can exist in extreme cold and extreme thermal vents. What are thermal vents? Have you seen that how geysers, they are heating or boiling the water? So that is more than 100 degrees Celsius temperature. So how they can exist? So this can be amazing, that how they exist under such conditions. The water from the hydrothermal vent is rich in dissolved minerals and supports a large population of chemoautotrophic bacteria. So students, if you remember, in class 11th, we have studied kingdom Monera. It is classified into two parts, eubacteria and archaebacteria. We have studied under archaebacteria that there can be two kind of bacteria. Some who can photosynthetically make their food and others who can use several chemicals for making their food. So here we are talking about, like, if a question comes in your mind that how bacteria survive under such extreme critical condition. So answer is they are using sulfur compounds such as hydrogen sulfide, H2S, which is a chemical highly toxic to most known organisms and produce organic material through the process of chemosynthesis. If I ask you why photosynthesis is not written, so the answer is because they are not using light as a source, but they are using chemicals for production of their food. Now the next question arises, do not they get killed under such condition? So bacteria have some amazing survival mechanisms. They are attached with their genome. Genome means set of chromosomes. So here we are talking about the internal nucleic acids which are acting as a genetic material. So there are certain group of enzymes and proteins which help them to overcome the adversity. All of you know what is adversity means? Those unfavorable conditions. Unfavorable conditions means if they are living in polar regions, means where there is extreme cold as well as where there is extreme heat condition. So they can produce both heat shock and cold shock proteins which protect the bacterial cell from extreme heat and cold respectively. Then they have talked about that there are diverse means different varieties of microorganisms which are found. Number one, protozoans, it can be amoeba, it can be paramecium, plasmodium. Then bacteria, as we have studied, coming under kingdom Monera. Fungi and microscopic plants, viruses. Viruses we all know as it has troubled us a lot. So viruses are there. The number two, viroids and also prions. So these are proteinaceous infectious agents. So if I discuss about viroid and prion, let's read. This is important. Viroids, they are infectious RNA molecules. They can cause several plant diseases. They are not causing any infection in animals, but they are only infecting plants. They can be transmitted like viruses. Then prions, they are protein molecules, but they are infectious. Here we are not talking that viroids and prions are useful. They are always infectious. Number second, they can cause either animal or human diseases. Means they are not causing any disease in plants. It can be insomnia. Insomnia means lack of sleep. It is not in general that you are lacking of sleep and it can be prions entered in your body. No. Insomnia may be leading to depression also. Second is mad cow disease. It may enter in the body of cow and lead to lower down its metabolism. Next is very resistant to disinfectants. Means if we use either sanitizer or we wash our hands with soap or any liquid detergent, there will be no effect that these prions will get killed. So their property is exactly different from viruses. Viruses can get killed by continuous washing our hands, but these are very resistant to disinfectants. Then next is exact mechanism unknown. So still there is a controversial case against these prions, that how they are made and how they are transmitted from one body to another. So next students, we will talk about microbes. They can be grown in nutritive media to form colonies. Colonies means group of bacteria, culturing them so that we can see them with our naked eye. Why it is useful? It can be useful in research, right? Next is, if you see this page, so many diagrams are given on this page. So we will start from number 1, that is figure 10.1, bacteria. All of you remember that we have classified bacteria according to their shapes. So if they are rod shape, they are called bacillus. If they are comma shape, they are called vibrio. If they are spiral, they are called spiral. And if they are spherical in nature, they are called as coccus. Then next is figure 10.2, in which different kind of viruses are given. Let's first of all study a bacteriophage A picture. In this is a virus. So I am repeating, bacteriophage is a virus. PHAGE means a virus. Then why it is termed as bacteriophage? Because it is a virus which can parasitize. Parasitize means it can enter a bacteria, any bacteria, either E. coli or any other, and then reproduce inside it. Bacteriophages are acting as those carriers which can be used in genetic research, or we can say genetic engineering. Like in chapter number 9, we have talked about several methods of plant and animal breeding. If it is performed artificially, means if specific traits are being inserted, how that insertion is done? That is done with the help of several bacteriophages. They are coded with that particular gene, and then they reproduce inside the bacteria, and that bacteria can be incorporated into either plants or animals where we want to get a desirable result. Then B is adenovirus, which causes respiratory infection. So this is a diagram of adenovirus. Last is rod shaped TMV, tobacco mosaic virus, which are causing several respiratory disorders. After this, figure 10.3 talk about colonies of bacteria and fungi. So they can be grown in laboratory. Petri dish you all are familiar with. So when they are grown, we can use them for human welfare. So let's study how are these different microorganisms used for human welfare. So let's start with the topic, very first part, how microorganisms can be useful in our household products. So this is not a surprise to us because we are using them every day. We are converting milk into curd. So that is with the help of microorganisms called as lactobacillus. What is the function of lactobacillus? They are the bacterias. Write down, these are the bacterias, and many other lactic acid bacterias. They grow in milk. How can milk converted into curd? Yes. So a starter is used, a small inoculum that we call as a jug, right? So a small curd is used that can be either called as starter, STARTER, or it can be called as inoculum, INOCULUM. Very small amount of that starter is added to, yes, can you tell me, it is added to cold milk or a lukewarm milk? Yes, you are right. So first of all, we are heating the milk. When it comes to lukewarm, it depends upon season wise also, right? So we are adding a starter, and what will happen throughout the night? Those bacteria will grow and then they produce acids. So can you give me answer which acid is produced inside it? Lactic acid, right? So that lactic acid, it will coagulate and it will partially digest the milk proteins. If I ask you a question here, if you have to choose from milk and curd, which one is more healthier to eat? So answer is curd. Why? Because those lactic acid bacteria, they improve the nutritional quality by increasing vitamin B12. And even when your stomach is not fine, means when it is upset, right? You have loose motions or there is dehydration problem. So we are usually intaking curd because those bacteria, they are very beneficial in playing role in digestion also. So this we have studied microbes in household products. So they are, first of all, we are talking about curd. From milk, curd is formed. How it is formed? Those bacteria, they coagulate the milk proteins, and finally we get a curd that is much healthier as compared to milk. Now coming to second part. Dosa, idli, these all are southern foods, right? So they are also fermented by bacteria. And you have seen when those dal and rice, they are being grinded, and what happen when they are kept for either 12 hours or 15 hours? Yes, the batter gets puffed up, it gets swollen. So it is due to the production of carbon dioxide gas. Now here the question is, can you tell which metabolic pathway is taking place resulting in the formation of carbon dioxide? So the answer is fermentation. There are two kind of respirations, aerobic and anaerobic. So anaerobic respiration means when there is alcoholic fermentation, ethanol is produced, right? As well as carbon dioxide is there. Then where do you think the bacteria for these fermentations came from? Yes, so it occurs when yeast. So yeast is required, right? It converts sugar present in flour. Which flour? Which we have added. So it changed that into carbon dioxide in ethanol. One carbon dioxide gas is trapped by gluten proteins. Where is this gluten present? Gluten is like a glue by which we can mix the flour with water, right? So which causes dough to rise. That dough gets risen up. Fermentation results in a light and airy crumb in bread products. So yeast is also used in fermentation. So bread is made by this method. So let's read name of the yeast that is used, and bacteria are also used. This yeast is also used. Saccharomyces cerevisiae. I am repeating how to pronounce. Saccharomyces cerevisiae is a yeast that is used for making dosa, idli, chole bhature. It is due to the production of carbon dioxide gas. So it is coming under anaerobic respiration. Then number third is a drink. A traditional drink in southern India is toddy. That is done by fermenting sap from palms. What is sap? The fibers of the palm tree are used. They are fermented. Fermentation, I am repeating, fermentation is alcoholic fermentation that is done when microbes are used and they are undergoing respiration without oxygen. There are several ways. Fish can be fermented, soya bean, bamboo shoots, and several different kind of foods are made. Last but not the least, very important, how to make a cheese. So cheese, there are different kind of cheese which are available. That is also made with the help of microorganism. How do cheese differ? They can differ in color, they can differ in texture, flavor, as well as taste. So everything depends upon which kind of microbe is used for making that particular cheese. Now, let's talk about number first is Swiss cheese. Propionibacterium shermani. Kindly listen. Propionibacterium shermani. This is used for making Swiss cheese. There are large holes in Swiss cheese due to larger amount of carbon dioxide. Board question can come, why do large holes appear in Swiss cheese? So answer will be due to the production of large amount of carbon dioxide, these large holes appear. Second, you can see Roquefort cheese. This is ripened by growing a specific fungi on them. A fungus is grown and it gives a particular flavor to this Roquefort cheese. So this was about these microbes, that how are they useful in household products. But now I also want to tell you that how cheese is being made. This is not in syllabus, but it could be interesting. You must know that how cheese is being made. So cheese production. First of all, we start with raw milk. You can see a container. Raw milk is there. There are two methods. One is traditional cheese making. Second is another method, mechanical method. First of all, pasteurization is written. Can you see pasteurization? Yes. Pasteurization means the process of heating milk up and then quickly cooling it down to eliminate certain bacteria. Do you all know how the packed milks are coming to us? It can be Amul milk, Verka milk. So when those packets are coming to us, you can read that it is written pasteurized milk. Means it is heated at a very high temperature and then suddenly cool it down so that it should not contain any bacteria and it is safe to transport from one place to another. And it is recommended also that there is no need to boil that milk because all the processing is already done. Okay. After pasteurization, it is standardized and filtered. And then you can see both traditional and the other mechanical methods are now same. That milk is now started curdling. Coagulation is there, right? All the milk proteins like the curd is formed. Similarly, that coagulation started. Now what is being added inside it? Number one, fermenting microorganism. We need certain microbes that can ferment it. Then inoculation, we all know. Inoculation is what? The basis of cheese making relies on the fermentation of lactose. Where is that lactose present? Write down, lactose is the sugar present in the milk. So it is fermented by lactic acid bacteria. So if the question comes in competitive exams, that which kind of microorganisms are used in cheese making, so that are lactobacillus bacteria or lactic acid bacteria. Then number third, what is being added? Rennet enzyme. Now here the importance of rennet enzyme is, rennet is a complex of enzymes containing chymosin, pepsin and lipase. This enzyme, where it is found? In ruminants. Have you seen how cows and buffaloes, they are sitting and chewing their cud, cud cud, right? So because they are called ruminants and they have this kind of enzyme, proteinaceous enzyme, so that they can digest all the food. So industrially, rennet is the main enzyme in cheese making, which is used to separate milk into solid curds for cheese making and liquid whey, right? So when that coagulation is done with the help of rennet and microorganisms, now it is being cut, it is stirred and it is cooked. Now what is done after cooking? That is going to put in a mill. Salt is added, right? Salt is a very good preservative, right? So salt is added, then cottage and cream cheese is extracted, then pressing is done, it gets ripened, time is required, and then ripened cheese is available to us. I hope you enjoyed this. So we have studied what? How microorganisms, they can be used in as a significant role in our household products. We have studied number one, curd making, number two, dough making, number three, toddy drink, that is a traditional drink in southern India. Then last we have talked about cheese making. Thank you.